Today's video I will discuss about structure and functions of liver. Okay, now then start. The liver is the largest gland in the body, situated in the uppermost part of the abdominal cavity on the right side beneath the diaphragm. It is about 1500 gram in weight. The weight of liver is 1500 gram. It is irregular in structure. The liver is in irregular shape structure. The upper surface is convex. The upper surface of liver is convex and lies beneath the diaphragm, and its under surface is irregular. Liver is mainly divided into two lobes. The liver is mainly divided in two lobes, and one is right lobe and another one is left lobe. Longitudinal fissure separated caudate lobe from the left lobe. The liver is subdivided into four lobes, that is the right lobe, left lobe, caudate lobe and the quadrate lobe. Okay, now see the structures of livers and the liver's lobe. That is the quadrate lobe and that is the right lobe. This part is the right lobe and this is the caudate lobe and that is the left lobe. Okay, now you just see the lobes of the livers, and then you will see that the artery vein and bile duct, it's the artery vein and it's the bile duct, it's the structures of TS of liver lobules. Okay, it's the central vein. Okay, now then the liver is made up of lobules which are polyhedral in shape. The livers made up of lobules which are polyhedral in shape. Each lobule is composed of cubical cell. Each lobule is composed of cubical cells arranged radially, and the vessels of the liver are connected by liver cells, the protoplasm of which contains many enzymes. Masses of such cells which form hepatic lobes are 1 mm in diameters and are separated from one another by connective tissues. The branches of portal veins, hepatic artery and bile duct are enclosed in a tissue covering called Gilson's capsules, which forms the portal canal. These portal canals are present surrounding lobules. The blood from portal vein comes in close contact with the liver cell. The small capillaries collect the blood and unit to form vein in the center of the lobule is called intralobular vein. The small capillaries collect the blood and unit of form vein in the center of the lobule is called intralobular vein. They pour their contents into another vein. Is called sublobular vein, which unite with the hepatic vein passing directly into inferior vena cava. Okay, then the bile is formed by hepatic cells and is secreted into fine bile capillaries which comes between the liver cell lying between the two cells and carried to the periphery of the hepatic lobules to intralobular bile ducts. The bile capillaries are separated from blood capillaries and hence the bile and the blood nor mix. Okay, the intralobular bile duct unit to form the hepatic ducts. The large bile ducts have lining of columnar epithelium and outer fibrous muscular tissues. By means of contraction of the muscular layer, bile is carried away. It is temporarily stored into the gallbladder. Okay, Now then, study the physiology of liver. What is the physiology of liver? Now, the first one is the first point of the physiology of liver is the secretion of bile, and the second point is glycogenic functions, and the third point is formations of urea, and the fourth one is desaturations of fats, and the fifth one is functions of related to blood, and the sixth one is the maintenance of body temperature. Okay, and the last one is excretions of toxic substances. Now we will discuss one by one in clearly. Okay, now the secretions of bile. Hepatic cells secrete an alkaline fluid. The hepatic cell secretes an alkaline fluid called bile. The bile is secreted from hepatic cells. Okay, it is composed of 90% water. The bile is composed of 90% water. Bile salts, that is the bile salts are sodium glycolates and the sodium taurocolates. Sodium glycolates and the sodium taurocolates is mixed, and that is the called is the bile salts, and the bile pigments are bilirubins and biliverdins. Okay, and then the cholesterol and mucin. The bile salts are helpful in digestions. The bile salts are helpful in digestions and the absorption of fats. The bile salts are help to digestion and the absorption of fats. They reduce surface tensions. The bile are reduce the surface tension of fats and help in their emulsifications. RBCs are destroyed in the liver and spleen. RBCs are destroyed in the liver and spleen. The hemoglobin of RBC is broken. The hemoglobin of RBC is broken down into globulin and hema. The hemoglobins of RCB are broken down into globin and hema. You all know in our previous video our blood, and I will discuss that part, and our RBCs, WBCs and platelets. You all know hema and globulin. And the hem is again broken into iron and porphyrin. Hema is again broken down and is broken down into iron and porphyrin, which forms the bile pigments later on. Okay, then the second one is glycogenic functions. Okay, the portal vein carries the blood from the digestive tract, which contains glucose and amino acids. The portal vein carries blood and from the digestive tract, which contains glucose and amino acids. It pours the blood into liver. The glycogenic functions of our liver. The portal veins carries the blood from the digestive tract, which contains glucose and amino acids, and it pours the blood into liver. The hepatic cells, by the actions of enzymes, convert glucose into glycogen, and it is then stored into liver. Whenever there is needed for energy to body, hepatic cells convert glycogen to glucose and pass it to the body cells through blood circulations. Okay, there is a need of energy to body. Hepatic cells convert glycogen to glucose and pass it to body cells through blood circulations. Thus, these glycogenic functions helps to maintain the sugar level constant in the blood. The glycogenic functions helps to maintain the sugar level constant in the blood. Okay, then the formations of urea. The portal vein carries the blood containing amino acids to the liver. The portal veins carries blood containing amino acids to the liver. The hepatic cells, by the actions. The hepatic cells, by the action of their enzyme, cause deaminations of amino acids. That means the amine group is set free, which forms urea, and urea is carried away to kidneys by the blood and is given out of the body through urine. Okay, you all know how to form urea. Then desaturations of fats, that is the fourth point of our physiology of liver. Okay, the digested fats in the form of fatty acid, the digested fats in the form of fatty acids and the glycerols are absorbed in the central lactal duct. The fatty acid and the glycerol again unite to form fats. This fat is carried away by the blood and is stored in the different tissues in saturated form. Whenever the energy is needed, this saturated stored fat is carried to the liver by blood. Okay, then the hepatic cells, cause the desaturations of this fat and pass to different tissues of utilizations. Okay, then what are the functions related to blood? And then the fifth point is the functions related to blood. The first one is formations of RBCs in fetal life. The second one is desaturations of RBC forming the bile pigments and irons. And the third one is the formations of plasma protein, that means the albumin and globulin. The fourth one is the formation of prothrombin. The formations of prothrombin and fibrinogens responsible for blood clotting. And the fifth one is the formation of heparin and a natural anticoagulant in the blood. And the sixth one is the storage of iron and vitamin B12. Okay, then the maintenance of body temperatures. Okay, as a number of chemical reactions occurs in the liver, heat is generated, which is helpful in maintaining the body temperature. The maintenance of body temperature, as a number of chemical reactions occurs in the liver and heat is generated, which helpful to maintaining the body temperature. Then the last one point of our physiology of liver, that is the excretions of toxic substances. The toxic substances entering the body through alimentary canal. The toxic substances entering the body through alimentary canal are destroyed in the liver. For example, alcohol, some drugs, etc. As the liver is responsible for a number of intermediate metabolic reactions. It is called the chemical factory in the body. Okay, then which is the chemical factory of our body? Which is the chemical factory of our body? You all know, that is the liver. Then the metabolism of our liver. When the nutritional materials which are end products of digestions are oxidized in the when the nutritional material which are end products of digestion are oxidized in the cells of the body, energy is released. All such chemical changes necessary for the fulfillment of vital functions of body are termed as the metabolic changes. Okay, then they are the sources of energy for activities of the body. And the minimum energy requirement per day is described as the basal metabolic rate, that's called BMR. Okay, this is the rate at which the sum of the metabolic processes takes place when an individual is at rest and has eaten no food. When an individual is at rest and has eaten no food for 12 hours, then the normal BMR values are in man is 170 kilojoule per meter square per hour, and in women it's 155 kilojoule per meter square per hour. Okay, the units of measurement of BMR are kilojoules kilojoules per meter square per hour. Okay, then you just see the chemical digestions of food. Okay, organs in the alimentary canal. Our alimentary canals have the organs are mouth, stomach and small intestine, and the digestive juice and enzymes are the saliva in the mouth, the saliva, and the secreted digestive enzymes is the salivary amylase. And then the carbohydrates are the cooked starch is convert into maltose, and proteins, mouth have no protein, no axons, and fats have no axons. In case of stomach, the digestive juice is gastric juice, and the enzymes are the pepsins, renin and gastric lipase. Okay, the carbohydrate, it's in case of stomach no carbohydrates, it's no axons. And then the proteins is the peptones, and the cousins is the paracousins, milk proteins, and the fats are the fatty acids and glycerols. And in the case of small intestine, the digestive juices are pancreatic juices, bile juices and intestinal juice. And the pancreatic juice having enzymes are trypsin and chromotrypsins, pancreatic amylase and the pancreatic lipase. In case of bile, it's bile salts and no enzymes, it's bile salts only. The intestinal juice is the peptidase, sucrase, maltase, lactase and entering lipase. Okay, the carbohydrate action on carbohydrate, its starch is converted to disaccharides, and the intestinal juice is secret, and it's the actions of carbohydrate is the disaccharides are the sucrose, maltose and lactose, and the sucrose is convert to glucose and fructose, and maltose is glucose and lactose is glucose and galactose. In case of protein, the peptones are converted into polypeptides, and the polypeptides is amino acids. In case of fats, the fats are converted into fatty acids and glycerols, and the unemulsified fats is converted to emulsified fats. Okay, the emulsified fats is the fatty acids and glycerols. The digestive juices volume or pH. Gastric juices volume is 1.5 liter to 2.5 liter per day, having pH is 1.6 to 3.2, and the intestinal juice volume is 2 to 3 liters per day, having pH is 7.5 to 8, and the pancreatic juices the 600 ml to 800 ml per day, having pH 4.5 to 7, and the bile, and in the case of bile juices 800 to 1000 ml per day, then the pH of bile is 6.8 to 7.7. Okay, you will study what is the structure of liver and also the functions of liver, and also you will know the chemical digestions of food. Okay.